Hello, 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 hello. Good morning. Good morning.
Morning.
Morning, ladies and gentlemen. You're going to hear this many times today, but I'm officially the first person to welcome you to IPPC 2023 in Lisbon. I'm Faraz Kamani. I head up global communications at PPTA. There's an amazing thing about Lisbon. It's one of the very few countries that has the same time zone as the United Kingdom, which is very pleasing for me. Yesterday, I thought we had the same weather as the United Kingdom as well, which was rather less pleasing. So I have to introduce a blanket of very quick things for you. First, I'd like to thank the sponsors for their generous support. Asahi Kasai, Fresenius Kabi, Haemonetics, Max System, Paysign, Synomed, Stradismed, Abbott, Kedrion Biopharma, and Immunotech Biocenters. Dinner tonight is off-site. Please check the app to know exactly where you're going. The shuttles will pick up the attendees at 6:30 p.m. outside of the lobby of the hotel. We encourage you to participate in the passport program, which is where you go around with a piece of paper and see all the exhibitors and get a stamp. And from that, you could win something magnificent, I believe. Also, please ensure that you're engaging with social media. We'd like to really push the name out there today. When you're here at IPPC, using our hashtag IPPC 2023. And if you've not already done so, please follow our social media channels. Another thing very important is be careful about your mobile phones. Perhaps put them on silent or switch them off. And if you don't have any other questions, if you do have any other questions, please reach out to a PPTA member of staff. And that's it. And I would then just like to introduce Maarten van Baelen, Executive Director Europe, PPTA.
Thank you very much for us. Ladies and gentlemen, it's an absolute pleasure to welcome you here today in Lisbon at the International Plasma Protein Congress. The timing of this meeting could not be more significant. As the EU moves towards the creation of a new regulation on substances of human origin, it offers us an opportunity to address the issues related to plasma collection in the entire European Union. Nevertheless, it has been quite a journey up to this point. All good historians know that without understanding our past, we may not be able to understand, never mind shape our future. Therefore, I'd like to talk a little about how our industry arrived at where we are today. How did we arrive at a period in which we are able to manufacture a wide range of life-saving plasma-derived medicines? How did we arrive at the point where we are able to ensure the highest level possible of donor safety and patient health? So where did the story of plasma begin? Blood plasma was first described by William Harvey. He was a physician to King James I of England. It was 1628. But the real understanding of the potential of plasma to treat human beings did not emerge until the 20th century. The Austrian-born immunologist Karl Landsteiner is known principally for his groundbreaking work in distinguishing the main blood groups in 1900. This actually also led to the groundwork for understanding plasma compatibility, leading to safer and more effective medical interventions. But there is another giant of the early 20th century that we should not overlook, the German physiologist Emil von Behring. He was a pioneer in the field of immunology. In 1901, von Behring received the first Nobel Prize in medicine for his discovery of serum therapy. This form of therapy was successful in treating the great outbreak of Spanish influenza in 1918, and eventually it was also used in the treatment of pneumonia, plague, and scarlet fever. Effectively, von Behring's work demonstrated the efficacy of plasma-derived therapies in combating infectious diseases. He inspired a new era of medicines. Now let us look into this new era. Plasmapheresis, initially carried out using animals, was originally described by Drs. Vadim Jurovic and Nikolai Rosenberg in 1913. The first studies of the effect of plasmapheresis on humans were carried out during the Second World War. The next big breakthrough for the sector also came in the 1940s. Professor Edward Cohn, working at Harvard laboratories, pioneered what was then the first practical large-scale method of plasma fractionation. This led to the development of essential therapies such as albumin, immunoglobulins, and clotting factors. His contribution has since saved countless lives and formed the basis of modern plasma therapies. Later in 1951, Dr. Josep Anthony Grifols Lucas conducted the first systemic study of the application of plasmapheresis in a number of plasma donors. They concluded that it was possible for donors to undergo plasmapheresis on a weekly basis without the quality of their plasma suffering. It also allowed for collection of a larger quantity of plasma when compared with the conventional method of recovery from whole blood. It is curious that it was at a congress here in Lisbon that Grifols Lucas met Edwin Cohn. While Grifols Lucas was presenting a safe technique for obtaining large quantities of plasma from healthy donors, Cohn presented a plasma fractionator, a device that separated out the proteins contained in plasma. These two major contributions marked the birth of a new industry, plasma fractionation. Another significant contributor to the field was the eminent surgeon Dr. Charles Drew. It is often said that he broke down barriers. I would argue, though, that he shattered a thick glass ceiling. At a time when racial segregation was rife in the USA, as an African American, Dr. Drew was instrumental in developing a method for safely collecting, processing, and storing large amounts of contamination-free plasma. He also developed techniques for transporting plasma, ultimately leading to the establishment of the first large-scale blood banks. His work paved the way for the efficient distribution of blood plasma products on a global scale. Ladies and gentlemen, all of this was a great breakthrough for rare disease patients. Dr. Judith Poole's discovery of cryoprecipitate in the 1960s was a turning point in the treatment of hemophilia. It led to the development of clotting factor concentrates that revolutionized the lives of patients with bleeding disorders. In 1961, Erwin Lepov and colleagues discovered C1 esterase inhibitor. The deficiency of this protein is associated with hereditary angioedema, which causes swelling in various parts of the body. Purified plasma-derived human C1 esterase inhibitor concentrate remains still the treatment of choice today. In 1963, Carl Bertil Laurel, a medical doctor and researcher working at the University of Lund in Sweden, discovered that an inherited lack of alpha-1 antitrypsin could lead to emphysema. In the late 1980s, alpha-1 proteinase inhibitor was purified. Starting with the discovery of A-gamma globulinemia in 1952, more than 180 genetic disorders were identified under the umbrella of primary immune deficiencies. The International Union of Immunological Societies today recognizes 9 classes of primary immune deficiencies, totaling approximately 430 conditions. Immunoglobulin replacement therapy continues to be recognized to be essential medicines for PID patients by the World Health Organization. Ladies and gentlemen, these are just some of the areas in which our industry continues to drive progress to treat rare diseases and to reach an ever-growing patient population. It is only by standing on the shoulders of giants, based on the discoveries of the past 150 years, that PPTA and its member companies continue to ensure that we remain committed to researching, developing, and innovating so that we can serve an even greater amount of patients in new therapeutic areas today and in the future. Thank you. Talking about the future, please allow me to introduce our new President and CEO, Anita Brikman, who will continue the opening address.
Good morning, everyone. Maarten, thank you so much for setting the scene with the magnificent legacy of plasma-derived therapies and the lives saved or improved as a result. This history is only one part of a broader tapestry. Every day, individual contributions and stories are woven in, making a profound impact on millions of patients who suffer from rare and chronic diseases. The well-being of these patients depends upon people like Evelyn Biegler. She's a marketing consultant in Germany. Zolt Horvath, a cybersecurity student at the University of Budapest. Dora Ivanka, she works as a bicycle courier in Hungary. And then there's Christian Koch, an educator who has donated more than 275 times throughout his life in Frankfurt. Their dedication has saved lives, and we need to celebrate contributions like this. In the short time since I joined PPTA, several people have already reached out to me sharing their personal stories, telling me about themselves or members of their families whose lives have been saved or improved thanks to PDMPs. I've actually also heard from individuals who donated plasma for the first time after they recovered from COVID-19. We know that the pandemic raised awareness of plasma's healing potential, but there is so much more work to be done and opportunities on the horizon. PPTA member companies are innovators and aim to deliver highly effective therapies to people around the world. Despite this, even in developed nations, some patients still encounter obstacles to access. To address this, we must multiply our advocacy and education efforts and address the needs for more widespread plasma collection. During my first week in this role, I visited our Brussels office and absorbed one major theme above all. The European Union is not able to provide sufficient volumes of plasma for fractionation to serve its own citizens. 446 million people struggle to collect enough plasma for medicines to treat their children, their parents, and their friends. But solutions are on the table. We have real-life examples showing exactly how it can be done. If the political aim is more sustainable and resilient supply chains for the manufacture of medicines, everyone must contribute to enhancing collection, both the public and private sectors. Here in the EU, we have Austria, Czechia, Hungary, and Germany, in which the public and private sectors work side by side to collect plasma. But still, as we know, there is significant hesitation to follow this path. Arguments referring to ethics have been raised, especially now when the new SoHO regulation is being developed at the EU level. But should we discuss the ethical considerations of compensating donors for their expenses and inconveniences without also shining the spotlight on the ethical aspects of saving patients' lives? There is no doubt that we face an uphill battle to encourage other European countries to implement more efficient collection systems based on plasmapheresis, compensation, and public-private collaboration. However, as I said, it can be done, and we have examples of it. How many private collection centers were present in Czechia and Hungary 25 years ago? None. Today, dozens of centers are collecting the starting material for the production of life-saving therapies. And we have an even more recent example in Ukraine, which reshaped its plasma collection system to better serve its citizens and security. This is a slow and incremental process, but there is progress that needs to accelerate to meet the rapid growth in patients' needs due to greater life expectancy, better diagnosis, and more therapeutic indications. We must continue our efforts to enhance access to plasma-derived medicines. To paraphrase John F. Kennedy, not because it's easy, but because it is hard. We, as PPTA, have a global impact. Candidly, I will tell you, it is one of the things that drew me to this association. We have a responsibility to the source, generous donors, and equally to our patients. The new PPTA logo embodies the patient-donor relationship. In our conference programming today and tomorrow, you will hear about our industry's commitment to donor health and safety. You will hear about the untapped potential of plasma-derived therapies, and you will hear from the patients who benefit from the unique nature of PDMPs. This focus on people lies at the heart of everything we do. PPTA and its members can never lose sight of that, and we can be proud of the advances we have made as an industry. Nevertheless, as you know, awareness about plasma donation has never reached the levels associated with blood donation. People simply don't know enough about it, nor how unique plasma-derived therapies are. We have to expand our impact. We need to elevate and celebrate our donors, and we need to amplify our voice as an industry. It is my commitment to all of you that doing so is one of my primary objectives in leading this association into the future. In closing, I'd like to share a story that moved me very much when I heard it for the first time. It is the story of a man who started donating plasma during his university studies, and he is still doing it. Today, any compensation he receives goes to charity organizations picked by his own friends on social media. And this man is among us today. Please join me in a round of applause for Tillman Häusler. Thank you, Tillman, for your magnificent dedication. Tillman will be sharing more about his story later during the Congress. My friends and colleagues, the story of the plasma sector is an evolving one. We are grateful for donors, dedicated to patients, caring for all. This is who we are. This is PPTA. Everyone, welcome to Lisbon. Welcome to the International Plasma Protein Congress 2023. Thank you.
It's an honor for me as well to welcome as well on stage Ferdinando Borghesi. We will continue the opening ceremony with an interview of him. Ferdinando is a Regional Commercial Director Intercontinental and responsible for toll fractionation and international contract manufacturing for Kedrion Biopharma. More importantly, he is also our EU Board Chair. And the interview will be conducted by Faraz Karmani, our Director of Global Communications.
Yeah, thank you very much for the speeches. Now I'm afraid you're just stuck with me again. So, Ferdinando, we often talk about from vein to vein, which is a running theme of this Congress, from the donor to the patient. But there continues to be a lack of awareness among the public regarding the importance and impact of plasma donation in saving lives. How, in this modern age, riddled with ever-increasing technology, do you think that we can make a breakthrough to address this type of issue?
Well, Faraz, I think we do have the opportunity to do it. And I think the lack of awareness was in the word of Anita and Maarten just a few minutes ago. It's still the much need of improving awareness. We have tools and we have expertise to do it. We can use our social media individually and commonly. We can use all the tools of the PPTA, regardless if you are members or not. So PPTA make broad communication to raise awareness. We heard from Maarten a few minutes ago that the primary immunodeficiency has almost 500 type of disease connected to it. And yesterday we heard in the stakeholder meeting from IPPC that recently additional 5 gene mutations were identified. I really challenge all of you to list those. I cannot do myself. But I'm shocking that this awareness is needed in the public domain with the scientists, with clinicians, with the regions, with the states that's still not implementing the newborn screening for kids to identify. I think there is a lack of awareness, but we are here just to raise this awareness and make better education to all our stakeholders.
Yeah, I mean, it's very interesting that you say it's just not a kind of Europe-wide thing, but that we have to go deeper and deeper into each individual
Correct.
Member state.
Correct, and this is not just Europe. It's an issue in Europe. You can believe in the rest of the world, where all the countries remains underserved.
Yeah, absolutely. So, moving on slightly from that and looking at what we're in the age of SoHO now in Europe, the Substances of Human Origin Regulation. What do we need to do to actually combat what I think is a wide variety of misinformation that's spread about our industry? How do we approach this?
I think we need to stay very, very focused on facts and evidence-based scientists, right? Mm-hmm. So we do have clear evidence that showed how the donation is safe and how our products are safe for the patient community. Anita clarified that the donors' health and donors and the safety of our patient community is at the center of all the manufacturers. And I think we need to continue to remark by educating and disseminating such an evidence. So we have a crucial role. SoHO is a huge opportunity for us to make and differentiate the unicity of plasma proteins and the dependency of the plasma as a source for producing our plasma proteins.
So essentially, it's a combination of communication and advocacy.
I think both. Yes, communication might help to disseminate, but we also need to leverage our patient community and the donors community to make this difference and trying to raise the voice of the countries that are now really in a coexisting system, public-private, to collect more. Those are tangible examples that there is the ability to do it.
And when we talk about collecting more, we know that as we stand today, there are only 4 countries in Europe that allow this kind of public and private collaboration in order to collect plasma, which is obviously beneficial for the entire EU. But what practical measures can we adopt to convince other member states that this is the right approach for the EU?
I'm a bit shocked because I believe that we didn't learn that much from COVID, right? So COVID is a clear example that the strong dependency of the US collection procured a clear need to collect more in Europe, right? We should have learned. It was not a decade ago, it was a couple of years ago, right? So I think this was a proof that the strong dependency of the US collection created a problem in Europe and the rest of the world. I think we need to stay still, focus on facts, cross-checking consumptions versus collection in Europe, and there's a clear understanding, even in Europe, where in theory there shouldn't be any issue in the access to care or diagnosis or awareness. There are still countries that have a much different consumption per capita on IG, albumin, and factors compared to other member states. I think we have to stay on facts. We need to share examples. And yesterday we discussed with some colleagues at the European board that we need also to simplify.
Yeah.
Our stakeholders, primarily the institutions, don't talk the same language. We probably need to simplify, state a clear message, and trying to make facts and the evidence on their hands to make able to make a decision, right?
And do you see, have you a feeling that there are certain member states who may come our way and actually allow this kind of partnership?
I think we are talking to Greece, we are talking to Croatia. I think there are some of the member states that can leverage their voice, even the existing ones. We named Germany, Austria, Czech Republic, Hungary. I think we're this coexisting. It's why we should think about one of the channel prevailing on the others. We simply need to collect more. And we have seen that through affairs, we can collect more and better.
That's right. Okay, somewhere slightly different now. We're wrestling at the moment with SoHO, but there's another thing coming on the horizon, and that's the pharmaceutical legislation in Europe. What do you see are the main challenges to our sector of this pharmaceutical revision?
I think that this was, if I remember right, 27th of April, the proposal was made available on both the directive and the regulations. I think there is a strict connection with SoHO. For the use of plasma as an active ingredient to produce the plasma protein medicines. So I think there are some priorities more than challenges that are linked to the management of shortage of any product, and this is one of the priorities in the proposal. There is the GMP timing and duration and the regulations around this. And for plasma proteins, again, is a managing of a possible event of shortage that this is important. Is long, I would say, long journey, because we expect after the election to 2024, the process would be more and more, I would say, active. We have seen how the MEPs have been componed for both directive and the regulations. I think we need to talk to institutions again, as we have done with SoHO so far, and we'll continue to do till the final stage. We have to continue to differentiate the plasma proteins because of the unicity we do have compared to the pharmaceutical, because we strongly depends on the active ingredients that is plasma again. So I think there's a strict independency with SoHO regulations, even with EU plasma legislation.
So basically, the way I see it is plasma, the plasma in the collection is part of the SoHO, and we move on to the medicines part, under the pharmaceutical legislation.
That's right. That's right.
Okay. So we have a lot of rare disease patients across Europe who depend on plasma and depend on plasma-derived medicines, but not all of them have that appropriate access. So tell me a little bit about the hurdles to access across Europe and how we as an industry can address this effectively.
First of all, so we are always used to talk about IVIG and albumin, but within plasma protein medicines, there are ultra-rare diseases that are not even well reported or diagnosed, right? Sometimes it's a matter of access, sometimes it's a matter of diagnosis or awareness, but and sometimes the access is limited by some, I would say, financial measures, so clawbacks, taxes. So I think we need to, again, it's a matter of education. I think few institutions knows that in the plasma economies, the cost of the plasma takes the majority, right? And it's differently, I would say the financials are completely different than pharmaceutical. A strong clawback might limit the ability, right, to make this deal sustainable. And at the end of the day, remember that the patient community is the one that will see this difference probably before than us, right? Yeah. I think we need also to stay very close and different. I came from pharmaceutical. I have strong experience in pharmaceutical. I've seen the difference in the way we talk to the patient community because we share the same goal, that is access to care. Yeah. And when certain country measures limit this access to care, this is a tangible proved by limitation of products.
Okay. Move along to something that is, I think is quite intriguing for me, because we're sometimes seen as a very, I would say, old industry, and especially compared to companies that operate in the field of oncology, CAR T cells and so on. So explain to us all about what is happening in terms of innovation in our sector.
Yeah. Somebody can believe that some of our plasma proteins are commodities, but this morning we heard about new evidence on albumin, right? That is the first product that was isolated during the Second World War, right? So today there was a proof done by physicians and prominent clinicians how we can still continue to innovate by acquiring new evidences on our products, right? The way we administer this product, the way the patients can self-infuse this product, and there are novel therapies approaching. We strongly believe that, especially in the replacement therapy, so there still depends on immunoglobulins. Mm-hmm. And we continue to innovate even in the way we deliver the product with much higher standard of safety, mm-hmm. For both patients and donors. And I think the way we engineer our manufacturing to be even more secure about the safety of our finished goods.
Okay, excellent. So, final question. How does our industry contribute to society from the donor, industry, and the patient perspective? What do we do to make society better?
When I joined Carrion, we were saying, okay, we are working in rare disease, and we are a rare company. Because how many companies there are in rare disease in the plasma space? Very few compared to pharmaceutical. The way we do the difference is to continue to improve access for life-saving products. Mm-hmm. And we treat rare disease. I think the patient community every day will see the difference of having access to those products. I think we can make the difference for the community by continue to increase the access as much as we can, to be really in the countries with the patients that need more right? Independent about the limitation of the access and independent about the challenge we receive from institutions, I think we have a crucial role in this segment, and for the community, we need to continue with our aims.
Perfect. Well, Ferdinando, thank you for that wonderful chat. Now we have to move on to session number 1, donor health and safety. Thank you.
She sends her apologies and kind regards. So I will do my best to try to fill her shoes. But I can tell you I will be wearing flats and not her normal heels. So if you just give us a minute here, we'll, as everybody's getting mic'd up, it's kind of like a Madonna concert, I guess. You got to put on the special headgear here. So. Just a few items too, just real quick. There will be a Q&A session at the end. I'm hopeful that there's an opportunity for you to ask the questions and not me. I do have a list, but I'd rather hear from you. There will be individuals walking around with microphones, so it's a little hard to see up here, so if you want to raise your hand or stand, however you feel more comfortable, we'll make sure we get a microphone to you. So. Kind of feel like President Joe Biden here. He's got like the teleprompters, and it's no wonder he mumbles through whatever he's trying to say. But all right, we've got all the participants here. Thank you for your patience. This session is called the Donor Health and Safety Insights from Industry Studies, Future Outlook, and Wider Perspectives. And I think it was mentioned earlier, this is really one of the cornerstones of our trade association, and member companies are trying to express to the rest of the world that we have been monitoring our donors' health and safety, and we continue to monitor them, and we ensure that the plasma donation experience is the best experience a donor can have, and we ensure highest quality and safety for the individual that does voluntarily donate plasma. So this session will be showcase the industry's commitment to assure and preserve the health of plasma donors in both Europe and the US. It will provide an overview of published scientific studies by PPTA members, as well as present an outline of ongoing and future industry studies. These will be put in the context of scientific research published in this topic already. The session will also address the new roles of the EDQM as an expert body in setting plasma donor health and eligibility parameters as part of the SoHO Regulation Proposal. One other reminder too, if you downloaded the app, the presentations are on the app, so if you're taking pictures, you don't necessarily need to take pictures because you already have copies of them. So I just wanted to get that out to you. I'd like to introduce our first speaker, Michelle Franson. She has been with the Plasma Protein Therapeutics Association since May of 2018. As a senior manager, she's lead for project and study management. She works with various stakeholders in developing, implementing, and executing the industry's donor health studies and projects. Prior to joining PPTA, she spent over 10 years as a Navy flight officer in the US Navy. As a civilian, she worked in women's health research and in occupational and environmental health consulting, conducting exposure assessments and causation analysis. Ms. Franson holds a Master's of Public Health and Environmental Health Science and Master's of Professional Studies in Leadership, Education and Development, both from the University of Maryland, College Park. She earned an undergraduate degree in exercise science. I've worked very closely with Michelle, and she's top notch, and she's done a lot for the industry and trade association. So please welcome Michelle.
Hi, good morning. I'll be giving today's presentation on PPTA's donor health study roadmap, the past, present, and future. As you've heard, Donor health is a top priority for the plasma collection industry. Globally, the industry has collected over 130 million plasma donations from voluntary remunerated donors over the past 5 years with an extraordinary safety record. A data-driven scientific rationale is needed to help inform and guide decisions regarding policies for plasma donations globally. In this presentation, I'll give a brief history of the PPTA Donor Health Initiative, talk about the roadmap, data categories, and studies in the roadmap. PPTA's Donor Health Initiative was a strategy to create this roadmap of donor health studies to be implemented by physicians and medical policy experts. The aim was to preserve study donation patterns and monitor donor health parameters under industry standard volumes and frequencies through plasma vigilance, which is maintaining donor adverse events, data collection and analysis, ensuring that the roadmap is adequately resourced and managed for successful completion, publishing those findings when they become available, and advocating to regulatory stakeholders for science-based parameters for plasma collection, such as the US FDA, European Commission, EDQM, and the EUMC. So what is plasma vigilance? In 2006, the United States Federal Advisory Committee for Blood Safety and Availability, also called BPAC, recommended that the federal government engage in efforts to enhance safety monitoring for blood products, cell and tissue products, and solid organs in partnership with private sector initiatives. These monitoring systems, collectively termed biovigilance, were viewed by BPAC as important tools for improving outcomes related to transfusion and transplantation therapy. In turn, PPTA created the International Quality Plasma Program, or IQPP, Voluntary Standards Program. By 2014, PPTA established the IQPP Donor Fluid Administration Standard and the Standard for Donor Education. Then, in 2016, PPTA established the IQPP Standard for Recording Donor Adverse Events, which was again updated in 2018. As these standards were specific to plasmapheresis, the term plasma vigilance was coined. Here are 5 categories of our roadmap of donor health studies. One is plasma vigilance that I just talked about. The other 2 are long-term health effects and short-term health effects. Long-term being what happens well after a donor leaves the donation center and over time. And short-term health effects, what happens to a donor right after they leave that donation center. Others are donor demographics. Who are the donors that come into our centers, and why do those donors lapse when they stop coming? Since 2018, PPTA has published 2 donor health studies that are part of this roadmap of donor health. The first one was the ferritin study, as you see on the left, which was frequent source plasma donors are not at risk for iron depletion, or the FLIP-D study, which shows that few US source plasma donors have iron depletion, and frequent US source plasma donation does not adversely impact iron stores. The second one was published in 2021, which is our plasma vigilance or donor adverse event study, which showed that the overall adverse event rate was 15.85 per 10,000 donations, and 90% of all of those adverse events were classified as hypotensive and phlebotomy events. You will hear a little bit more about these 2 studies with Dr. Darmont's presentation. Currently, or just recently, last month, we submitted 2 studies for publication, which are currently under review. The first one was the effects of donation frequency on US source plasma donor health, which assesses self-reported functional health and well-being of US source plasma donors in 4 donation frequency groups, including effects that may be associated with impaired immune function over different time periods. The second one was why do US source plasma donors stop donating? This explores source plasma donors' self-reported motivations for donating and reasons for a lapse of 6 months or greater. And these, the study participants were part of the previous, or the effects of donation frequency on US source plasma donor health study. Currently, we have 3 donor health studies that are currently in the works. The first one is the hypotensive donor adverse events. So this is looking back at our previous plasma vigilance paper, which had 4 months of data, looking at hypotensive focused adverse events. And then currently we're doing a 12-month donor adverse event study. The member companies collected data from January 1st, 2022 through December 31st, 2022. And they're currently in the process of uploading that data to review a 12 months worth of donation history with the hope that we can look at the rare adverse events, do analysis on that, and look at seasonality and or geographic trends with adverse events. With that 12 months of data, we're also going to be looking at donor demographics. Who are our donors? How often do they donate? And so on and so forth. In the future, we have a short-term health effect study and a long-term health effect study. The short-term one will be looking at the immediate effects post-donation, to look at the ability to exercise and concentrate on tasks at work or school or other variables following donation. A preliminary timeline would be 2024, Q4, working on the protocol, and then recruit for the study, and then do the analysis by 2026. Long-term, we look at a longitudinal donor health study, which is a prospective study that will follow approximately 10,000 applicant donors for 5 years. We'll be working, hopefully, with external people to help develop this protocol, and then by 2026, if the protocol is developed and the feasibility is good, we will start recruiting for this. So in conclusion, PPTA's donor health study roadmap demonstrates PPTA's and its member companies' commitment to assure source plasma donors' health and ensure that plasmapheresis procedures remain safe. Thank you.
Thank you, Michelle. We're very fortunate with our next speaker to have Dr. Ramona Darabont, who is currently the lead physician at BioLife Operations, Takeda. She started out as an independent contractor for Baxter 7 years ago. She also works as a family physician in Austria since 2012. She carefully supports and motivates multilingual donors before, during, and after plasmapheresis. And she ensures best possible product quality and safety. So please give a warm welcome to Dr. Darabont.
Thank you. Okay. Hello, ladies and gentlemen, esteemed colleagues. As Robert already told, I'm family physician. Also, I'm a head physician in a plasma donation center, and I'm a very lucky girl. I can care and work with plasma donors and also my patients. So I have the unique opportunity to take care of my sick patients, where I also see the need for plasma-derived therapies, and I also see the donors and care for the donors, which now I have the both sides of plasma therapy. On the one hand, the raw product from my very nice donors, and I have the patients who receive their therapies, and their life quality goes up. So the donor health is a top priority in the plasma industry. Yes, of course. The health of the donor should not be compromised by their donations. I'm also bound to the Hippocratic Oath that first, do no harm, second, be careful, third, heal, if possible. So you see, the donor health is really an imperative theme and topic during all the plasma donations and collecting of plasma. The overview of my presentation, I will show you the worldwide regulations for plasma donations. And I will highlight some studies from the EU and the US with the topics of long-term plasmapheresis, donor adverse events, iron stores, bone health, blood pressure, cholesterol, and the IPPC standard. So here you see a very neat presentation of some countries in the world, mainly in the EU, where you can see that the 4 countries, Austria, Czechia, Germany, and Hungary, allow compensation for donors. You see also the difference between the donor frequencies per donor and the allowed donation amount with each donation. So you see in Austria and in Germany, we have 50 times donations per year, as well as in Germany, 60 times a year. We also have the USA and Canada and Austria, where you see the donations differ greatly. Okay. Now to the EU regulations. Due to the lack of scientific data in the 90s, European regulations on plasmapheresis were based on the best knowledge and practice in transfusion medicine. Since then, a lot of studies came out. There has been a scientific knowledge that has advanced with the key parameters of volume per donation and frequency, the monitoring of donor adverse events, and safety in laboratory in medical care, as well as in the documentation procedure. Here you see the EDQM blood guide, the 21st edition, the quality and the safety standards for collection, preparation and testing, and use of blood components based on the latest scientific developments. You see here there has been a draft coming out, and the substance of human origin regulation proposal is that for those standards concerning donor protection or elements thereof for which no implementing act has been adopted in order to supply such as standards or elements thereof, substance of human origin entities shall follow. The important is, though, that this EDQM blood guide will be implemented into the donor protection concerning other than transmission of communicable diseases. Here you see the proposal for apheresis donation. The donor should be informed of the risk associated with the apheresis procedure, of course. We do extensive donor education before letting them donate, and they have to give consent before each donation. The proposal is that the plasma collection centers need a responsible person, or at least a physician, which can change with national and regional regulations. The maximum allowed plasma donation frequency is 33 times per year, at least 1 week apart. The minimum of immunoglobulin G is 6.0 grams per liter, measured either annually or every 25th time donation, and the donation volume with coagulation should be 883 milliliters maximum. The national and regional regulations could be based on total blood volume estimation and or body mass index. The total volume of all components should not exceed 16% of the total blood volume maximum. So what are these? What is the evidence for these parameters? You see here in 2004 there has been made a study effects on plasmapheresis on humoral and cellular immunity, red cell count, iron metabolism, and cardiovascular risk markers. In this study, there were included 4 groups of donors, well, at least 3 groups of donors and a non-donor group for matching the results. They either donated 37 liters, 16 liters, or 10 liters as median annual net plasma donation. True is that all donors had significantly lower total serum protein, albumin, and IgG, than the non-donors, but the intensity of their plasmapheresis had no effect on these markers. All plasma donors had normal humoral and cellular immunity, so you can see that all their blood work came out within the parameters. There was no increased rates of iron store depletion, no increased cardiovascular risk. Then we have several studies on safety of long-term plasmapheresis. They all speak kind of the same language. In this study from Bergdorf, 2005, there were included 72 German donors who switched from moderate to intensified plasmapheresis and were followed up for 3 years. At the baseline, donors had significantly lower total serum protein, albumin, immunoglobulin G, hemoglobin ferritin levels than gender-matched and age-matched non-donors. The total serum protein, IgG, hemoglobin ferritin, transferrin, and cardiovascular risk markers and parameters of blood coagulation did not change significantly during the observation period. So subsequently, intensive plasmapheresis over 3 years did not impair the individual's ability to donate. This is also an important point that they are making. They looked up the dropouts from this study, and most of them were because of socioeconomic reasons and medical reasons not related to plasmapheresis. Very few of them had lower than threshold total serum protein. The CIPLA study. This is a very important one from 2006. It's a very large study including experienced donors. These donors had weight-matched donor volumes, and the serum protein, hemoglobin were determined at each donation, immunoglobulin at every 5th donation. Dropouts, like before, were mostly socioeconomic, medical reasons not related to plasma donations, and 16% of the dropped out had lower than threshold levels of IgG, serum protein, and hemoglobin levels. There have been in 5 subjects severe clinical adverse events related to plasmapheresis. The incidence in severe cardiovascular disease was lower in donors than in general population. This is very important because we see here that the plasma donation can be beneficial for the donor population, not just the cardiovascular concerns, but also, I will be talking about it later, for blood pressure and hyperlipidemia. The risk factors that led to dropping out as a result of low protein, low IgG and hemoglobin levels, the risk factors included young age, female gender, and low initial IgG levels to start with, as well as high frequency in donations. Body weight was not associated to cease the donation, and females and males with the lowest body weight were not at higher risk of dropping out. So the long-term intensive donor plasmapheresis under conditions investigated showed that, that plasma donation is a safe procedure. The CIPLA-2 study from 2013 included first-time donors and inexperienced donors as well as experienced donors. In this case, you see 1,284 males and 1,095 females were included. The weight-based volume of plasma donation is a little bit different from the CIPLA-1 study. And they donated also 60 times a year, like in the first CIPLA study. IgG and total protein levels were detected at least every 5th donation, and these levels had to be within the German guidelines. The results. So the average donation of plasma volume per donor on average were 19.1 liters. In the first CIPLA study, it's 43 liters. IgG levels below threshold were more found in females than males, 30.9% in females, 19.9% in males. Okay. Also, more IgG levels below acceptance were found in women than in men, to start with, and IgG levels did not predict dropping out, nor did the total protein. In the most frequent dropouts, the hemoglobin and the hematocrit were outside the threshold. So, further results now for the CIPLA-2 study, they also looked at side effects, which were not so many. Local reactions were 27.2%, technical problems 68.6%, and systemic reactions 4.2%. Most of the reactions were mild. Males had a significantly less hypovolemic reaction than females, of course. Blood pressure before and after donation did not predict the occurrence of side effects. Most dropouts were also caused by socioeconomic reasons and grade 3 side effects. This means that the donors needed major medical intervention or hospitalization. And the donors weighing 50 to 60 kilograms were at higher risk to drop out. Okay, there were no category grade 4 and 5 severity side effects. Okay, so you see in the CIPLA study, both of them had the majority of participants had a higher than maximal allowed annual amount of plasma donated than in the German guidelines. Both studies found that the regime is safe even when you include first-time and inexperienced donors. It is safe. So we come now to the IPPC standards for recording donor adverse events. I will show you the classification just in a minute. The donor adverse events recording, the standard for it provides the common language in the source plasma industry to classify adverse events. It's easy to use, it's objective, and the definitions are simple. The standard was piloted 2016 and applies to all facilities that collect plasma for manufacturing plasma-derived medicinal products. You see here there are a few categories and the recording requirements to it, including subfactories. There's, of course, most the hypotensive event, vasovagal or hypervolemia, major cardiovascular respiratory events, local injuries related to phlebotomy, citrate reaction, hemolysis, air embolus, hyperventilation, allergic events, immunization, or other events. You see some of the milder, uncomplicated, issued do not need recording, but everything else does. So to the plasma vigilance, the study that Michelle mentioned before, from 2021 by Schreiber and Michelle, the donation data was including 1.1 million donors, making over 12 million source plasma donations in a period of 4 months. The adverse events were classified via the IPPC donor adverse events, like I showed you before. 90% of these adverse events were classified as hypotensive and phlebotomy events, each at 8.32 and 5.91 per 10,000 donations. They were overall, the adverse events, 15.85 adverse events per 10,000 donation. Risk factor, as said before, was young age, lightweight, female, and first-time donor. So you can see here on the graph, that hypotensive phlebotomy events were the majority. Okay. Now we come to the red blood cell loss. The study has been made in 2013 and was incorporated into the donor fluid intake after the donation, the IPPC standard. There have been two groups. In a comparative examination in German donors, group 1 had the disposable tubing rinsed with and re-infused with the saline infusion after the donation, and the second group did not have that. You see, group 1 had an average ferritin concentration of 33.25 microgram per liters, and group 2 had 23.01 microgram per liters. You see, there is a big difference. And if the donor donates 45 times per year without rinsing, the result is a loss of 11.01 milliliters per donation, which hikes up to almost half a liter per year. When the rinsing is done, the blood loss by plasmapheresis can be reduced to 5 milliliters per donation or to 58.01 milliliters per year. So there you see, it's the donors do not lose so much blood as without rinsing. So for the plasmapheresis on ferritin levels and iron stores, this study is from 2018 by Schreiber. The total, this is an US study, and US donors were enrolled into 4 frequency groups by donation in the prior 12 months. No donation at all, 1 to 24 donations, 25 to 69, and more than 70 donations. Ferritin levels were determined for each donor. Donors with ferritin levels of less than 12 nanogram per milliliter were classified as having absent iron stores. The IPPC donor fluid administration standard, which was piloted 2014, administers a minimum of 250 milliliters of saline solution intravenous as a part of automated plasmapheresis procedure. The results. So compared to new donors, ferritin for females was higher in each successive frequency group. For 70 or more donations, ferritin was 13 nanogram per milliliter higher than in the new donor group. Compared to new donors, high-frequency donors had lower ferritin levels, like 114 nanograms per milliliter versus 100 nanograms per milliliter. Absolute absent iron stores occurred in less than 1% of all male donors and 4% of all female donors. So we come now to the bone health. The citrate infusion for the plasma donation in this study from 2017, the Sweden population was included into a cohort study for 67,970 females and 72,319 males apheresis donors, and they were followed up for 23 years. There was no association observed between apheresis donation and the frequency of risk and fracture risk. The incidence rate ratio in donors with more than 100 cumulative apheresis donations was 0.99, compared to donors with 9 to 24 donations. So there is no risk of fracture with high intensity donation and higher frequency. The cholesterol levels in this study by Rosa Bray from 2013. This is also a US study. 407 males and 256 females were included, and they were used across 16 weeks period with a high range of age from 18 to 69 donors. It has shown that there was a significant decrease in total LDL cholesterol in both genders at the baseline cholesterol levels, and that the greatest cholesterol decrease were associated with high baseline levels and 2 to 4 days between donations. Greater than that, there was no significant event. And also to be noted, there was a small but statistically significant increase of HDL cholesterol predicted for donors with low baseline events. So we can also help our donors who suffer from hyperlipidemia. The same the same study group for donors followed up also on their hypertension, the systolic blood pressure and the diastolic blood pressure shows that donors with baseline stage 1 or stage 2 hypertension profited from the plasma donation. They had marked decrease in systolic and diastolic pressure. Donors with normal blood pressure or pre-hypertension, there was no change in their blood pressures. So as a last point, I want to talk about therapeutic apheresis and plasma donation. In the draft, I got the feeling that the EDQM blood guide takes the concerns and side effects for therapeutic apheresis from there and translates it to the plasma donors, which I think is not quite right to do. The therapeutic apheresis is meant for chronically and or severely ill patients and subjects. The procedure is quite similar, but not always. There is a selective apheresis and an unselective apheresis. Selective for autoimmune diseases and unselective for familiar hyperlipidemia that cannot be treated otherwise. Heparin is mostly used as an anticoagulant agent, and the patients receive colloidal plasma expanders like gelatin during the apheresis and plasma, fresh frozen plasma, after the apheresis. So the common side effects and complications vary a bit. The vein puncture issues are there, of course. The hypervolemia is there with fainting. There is a possibility of heparin-induced thrombocytopenia, allergic and immunological reactions to fresh frozen plasma. In the plasma donation, there are only healthy donors, which are resilient donors, which means they can easily bounce back to their norm, whatever the circumstances. Citrate is used in the anticoagulation, and the donors receive at least 250 milliliters. It's mostly up to 500 milliliters of saline infusion after the donation. Common complications, vein puncture issues, as well as low blood pressure. So the conclusion of this is the scientific studies have shown there is no significant adverse effect on donor health by long-term intensified plasmapheresis if hemoglobin, total protein, and IgG levels are monitored regularly. The risk of adverse events decreases with the number of individual donations, and the cardiovascular risk of donors remain unaffected by plasmapheresis. Individualization of the individual IgG level status with begin of the donation may be considered to not to defer donors with lower level. The plasma industry is, of course, committed to protecting donor health and the safety and minimizing any potential risk. Here are the references for the studies. It is also on your presentations on the phone, so it will be easily to read it there. Thank you so much. Thank you.
Thank you, Dr. Darabont. I'm sure that'll stir up some conversation here at when we have the Q&A session. I have a few questions that I know or points I want to make with that. So, excuse me. Our final speaker is Dr. Rainer Moog, who is the Regional Medical Director of Octapharma Plasma and is currently the president of the German working group Plasmapheresis. He is a specialist for transfusion medicine and received board certifications in transfusion medicine as well as hemostasis and thrombosis. Professor Moog was educated at the University of Essen in Germany, where he completed his doctoral thesis and his master's thesis for transfusion medicine. He became associate professor and subsequently interim director of the Institute for Transfusion Medicine at the University Clinics Essen, Germany. Please, a warm welcome for Dr. Moog.
Ladies and gentlemen, Chairman, thank you for the kind introduction, and thank you also for the organizers for inviting me to this meeting, to this lovely city of Lisbon. It's really an honor for me to be here and to present our data about donor safety of individualized plasmapheresis, focusing on the IgG. This is my conflict of interest. And the background of our study was that we wanted to investigate adverse events of personalized plasmapheresis and to especially investigate the effects on the IgG level. And we had also a modification of the German chemotherapy guidelines in 2017. With a modification that there was a permanent deferral of donors in the case of 3 donations with an IgG level of less than 6.0 gram per liter. And that's why we asked, why that? And we wanted to show the impact of the IgG level on plasmapheresis and to show our asking, is this deferral correct or not? Now, I'm going to report on 2 studies which our group published in 2022. The first one on the left-hand side was from the IPS study. There were more than 20,000 donors recruited, and all adverse events were analyzed in this study. And on the right-hand side, you can see the paper which I'm focusing on with the IgG level. We analyzed this in a prospective controlled multi-center study. Now, what is the IPS study? The IPS study is an individualized plasmapheresis study which is based on the donor's IgG level and on the donor's body weight. And the aim of the study is donor safety. Why donor safety? The answer is very simple. We want our donors to come back and that they feel fine at our donation center. Now, this is the study design of the IPS study. It is a prospective, controlled, stratified, multi-center study. The stratification is in 9 study arms, depending on the initial IgG level of the donor and the body weight. Of course, we have included a control arm. 12% of our study donors are in this control arm, and they are donating according to current German chemotherapy guidelines. Up to now, there are 13 centers participating in the study, and we are going to increase this. And the study started in September 2007, and the donor may be in the study for up to 12 years. There is a continuous monitoring of all adverse events with respect to donor safety, and the study is monitored annually by a safety committee, and of course, the study was approved by regional ethics committees of the Chamber of Physicians. And it is very important to point out that it is still an ongoing study. Now, the study design. Let us start with the last row, with the control group. And you can see that up to 60 donations are possible per year in Germany. And the donation volume is adjusted to the body weight from 50 to 60, 60 to 70, and more than 70 kilograms. And now the 9 study arms. Of course, we have the body weight, as I just mentioned, and on the left column you can see that they are stratified also by the IgG for 3 groups, 6 to 8, 8 to 9, and more than 10 gram per liters. So when we talk about individualized plasma donation, you have to know that there is a correlation between, on the one hand, the initial donor IgG level, the donation frequency, and the adjusted stable long-term IgG level. And this example shows you the IgG management and how we manage it at our centers. So every resident has to learn first how the IgG level behaves. And this is just a typical example I want to show you. You can see at 0, the donor starts with an IgG level of about 13 grams per liter. And after 5, 10 donations, the IgG level goes down. And usually it remains at a very stable level. And that is a very important message for the donor. The doctor talks to him about these values and what is the optimal donation program for him. Now, the donor disposition of the IPS study as of June 2019, we start again with the control group. On the left-hand side, there were about 2,000 valid donors in the control group. Just to mention again, they were not stratified by the IgG. There were about 60% of withdrawals. There are 762 donors still on the study. Some are in the initial phase, and of course, about 1/3 are in the extension phase. And now on the right-hand side, the individualized program groups with about 70,000 donors valid with the IgG analysis in the population. And as I have shown you before, with the IgG levels in the low, medium, and in the high group. Also here, we have a relatively high number of withdrawals, and I can tell you this is quite normal. We received a letter to the editor to our publication pointing on this issue of dropouts. And as you have seen by my first speaker, most of these dropouts were due to socio-economical reasons. That's to say, the donor moves to another city, the donor has a new job, he has no time. There are no medical reasons why these donors drop out. This is very important. These are not clinical studies with the patient lying in the bed, and you can follow up him easily. So now I come to the paper published in Transfusion Medicine and Chemotherapy, which is called Sub-Study Design. Again, control arm on the left-hand side, and the individualized on the right-hand side. We have 3 or 4 groups of donors with different IgG measurements below the threshold. and they were analyzed in this subgroup. Sorry for this. And in the individual arm, about 14,000 wellness donors were included, about 11,000 with less than 3 IgG measurements below the threshold, and about 3,000 donors with more than 3 IgG measurements below the threshold. Now, this is the background of the IgG sub-study. We were concerned, wanted to analyze the incidence of infections. Our hypothesis was that IgG deficiency may result in increased risk of infections. You may know we use the IgG for treatments of patients with immunodeficiency, and we want to look at our donors with their IgG levels if it has an impact. You know, immunoglobulins are an important part of the adaptive humoral immune system, and IgG is the most abundant immunoglobulin in terms of quantity, the normal range being from 7 to 16 grams per liter. Now, we wanted to show in our studies that there was a non-inferiority of donor safety in donors with 3 or more donations below the IgG threshold compared with donors with none, 1 or 2 donations below the threshold. And as a key parameter, we use the incidences of adverse events. This is the degrees and the description of the severity of adverse events, ranging from mild over moderate, severe, life-threatening, and lethal. This is the standard grading of adverse events. And of course, we want to look at the causality of adverse events and plasma donation, and there were 4 categories from no relationships over without temporal relationship, possible and probable relationships. This is all the doctors had to document when following up an adverse event. So for statistics, we used descriptives. We use this mean, standard deviation, median, range, frequencies, and incidences. And we calculated the donation-based incidence, which was done as follows. Number of adverse events divided by the number of donations multiplied by 100. So I now come to the results. First, the ongoing IPS study. You can see the donation-based incidences of the control group and the study group, which were almost similar, and the related adverse events also almost equal between the 2 study arms. And most of the adverse events we observed were mild or moderate. Most frequent were events related to venipuncture. It was about 47%. And as I mentioned before, the majority of withdrawals was due to non-medical reasons. This slide shows you the number of severe life-threatening or fatal adverse events per year at risk. And you can see that the possibly or probably related events were very, very low in the study arms reported. Now I come to the IgG sub-study results. First, the control group. There's no slide here. Okay, I'll do it without pointing. On the left-hand side, you can see the number of IgG below the threshold, the 4 groups, and then there are the demographic data about the age at study entry and the number of donations per year. and the time at risk. This is also very important. And you can see that for the first three groups, these were all almost similar, and only for the last row with more than 3 IgG measurement below the threshold. Of course, you can see because these donors were longer in the study, they had longer time at risk, of course. Now, first, the adverse events without infections. There were 19 recorded. 3 of them were severe. There was only 1 adverse event with a causal relationship, and we found no adverse events of severity 4 and 5. So this shows you the 19 cases on the left-hand side here, and the calculated incidences of this. Okay, now it works. And this is just a summary again. And please note that only for the group with more than 3 IgG measurements below the level, there was 0.002 incidence of severe events. Now I come to the infectious adverse events, which of the severity 1, 2, and 3. And you can see that these were almost similar here for the 4 groups. And we observed no severity of grade 4 and 5. And these are the data for the control group. With mild and moderate infectious adverse events. Here, and only for the last group with more than 3 subsequent IgG levels below the level, you find higher incidence for mild and moderate adverse events. So, to summarize, there were nearly 100,000 donations during the observation period, and the time for the donor at risk was more than 3 years. In total, 187 infections were observed, 63 mild, 124 moderate, and our calculated incidence was 0.192%. There were 2 flus with a possible relationship to donation, and most frequent infections were influenza, cold, urinary tract encephalitis, and respiratory infections. And I show you these slides with death in plasmapheresis donors. Unfortunately, we observed 8 deaths during the study period. And I wanted to point out that we observed no malignancies. This is a very important issue because we noticed in the commentary of the EDQM that there was a relationship with plasmapheresis and malignancies. From our study data, we cannot confirm this at all. That's not right from our point of view. And this is a very important message for the auditorium, I guess. So, to sum up, there was no causal relationship in donation safety in donors with IgG levels below the threshold and those of the control group. And we think that this is a proof of concept of donor stratification by pre-donation IgG levels. Just to two US studies. The studies on the left-hand side has been already cited by my four speakers, but I think it's a very important study. Be more than 12 million donations were analyzed. You could see the results on the slide before. And on the right-hand side, this is a study by Dr. Hartmann, which says that we are not alone in Germany thinking about personalized collections. This was the impact study, that means improving plasma collection, novel plasma program with personalized target volume calculation. And by this means, they were able to increase the volume of the plasma collected without any significant side effects. So this is just a short outlook what we are going to do with the IPS study. The study is not finished. Our aim is to include 30,000 donors. And of course, we want to publish our study results. As it is an ongoing study, we expect a final publication in about 2035. And what we are doing now is to publish sub-results or to analyze them. That means a long-term follow-up of dropouts. And of course, we want to use our data for discussion with national and international authorities, thereby updating the guidelines of plasmapheresis according to the state of the art. So, to conclude, personalized plasmapheresis is feasible and safe. Frequent plasmapheresis with IgG monitoring is safe. The clear message is it won't make you sick. And donor safety is not compromised in donors with more than 3 IgG measured and below the thresholds. The incidence rate of infections in donors with low IgG levels were almost similar to other studies. And by doing this, individualized frequent plasmapheresis, we are able to increase the plasma support and supply and the self-sufficiency of Europe and our countries. Thank you for your attention, and I'm ready for your questions.
Thank you, Dr. Moog. If there's questions, I don't know who has the microphones. I can't see out there, but if anybody has any questions, but sorry. Oh, okay. Thanks.
Yeah, Roger, it's Matthew Ochko, Marketing Research Bureau. Question is for second speaker about the CIPLA-1, CIPLA-2. I noticed that the IgG level below the threshold was like 16% in one and 25 to 30% in the other one. So I was wondering, is there an explanation for that? Just different study protocols or something else? Because that seems pretty dramatically different.
Yes, thank you very much for the question. The IgG levels seem to differ because in the CIPLA-1 study, they had only experienced donors, which means their IgG levels were a bit lower already, like Professor Morg stated. After the first donation, the IgG levels sink with a frequency of donation, then they remain stable. And the CIPLA-2 study included first-time donors, inexperienced donors, as well as experienced donors. So there's a greater variation.
Thank you.
Thank you very much.
Josh, do you have a question, Dr. Penrod?
No, Roger, just Josh waiting.
Oh, okay. Well, it was quite a whirlwind of, a little over an hour of all the studies that PPTA and member companies have done. and continue to work towards in terms of focusing on donor health and safety. I mean, one of the things that I picked up on, and maybe we can have the speakers talk to, is the SIPLA study and the study that was done in the US for donor adverse events clearly demonstrate the risk of an adverse event to be a significant adverse event to be extremely low, extremely low. The vast majority of them were either plasmapheresis or phlebotomy related or hypotensive. And so I guess the question I raise is how, during this discussion with SoHO and the whole discussion regarding classifying a donation as high risk, whether or not these studies should be taken into consideration, right? So I don't know if you have anything to comment on or feedback on something of that nature.
Yes, thank you. My opinion is that the SoHO discussion and the EDQM blood guide falsely conclude that apheresis is apheresis, either it's therapeutic or it's a donation. Clearly, they think it's the same procedure, so it has the same effects on everybody, which it doesn't. So the donor adverse events are, of course, they cannot be left out for the safety of our donors. But it is also important to know that these severe donor adverse events are clearly a lot less than usually would be expected, even if it's about therapeutic about therapeutic apheresis. The side effects are not as high as to be expected. Also, if it's considered that therapeutic apheresis is about sick patients who do not have a resilience, who do not bounce back if we change anything in their system, who already come with a dysregulation. The donor adverse events are really a minimal risk for donors, and we do have extensive donor education in place before every first donation, so the donors know exactly what to expect and how to expect it. Also, they have the possibility to always come to the doctors and to the nurses present in the plasma donation center, we are all up to date in lege artis, so our care for the donors is the best we can provide with our technical and our possibilities in evidence-based medicine.
Dr. Moog, do you have any
May
comments?
I just add something? This is really very important that the donor is well informed about the procedure of plasmapheresis. We do that and inform them in detail at a talk, and we require written informed consent that he has understood what we are doing, what are the possible side effects, and how to avoid them. And it's very important to inform them that he has to be well prepared for the plasmapheresis donation. That's to say, to drink a lot before and to have a meal, to feel well, to come only to the donation center when he is healthy, and that we will assist them by avoiding the loss of plasma by doing a saline compensation at the end of the procedure. And by doing so, the incident rates of adverse events will be very, very low. And as I mentioned, we consider plasmapheresis to be safe.
True. I can second that.
Thank you. Don't forget, there's opportunities, microphones on either side. We have a...
It's possible?
Yes, please.
I'm asking for saline. I'm coming from the anesthesiological and intensive care side, and saline leaves the vascular system in under 20 minutes. And it is still used instead of ringer lactate or ringer solution, which are mostly staying about 2 to 4 hours. In the Schreiber study, besides venopuncture is hypertensive problems, the most percentage of adverse events. why we don't change from saline to ringer solution or other more potent fluid replacement.
Thank you for the question. It is important that we use the saline infusion because of the citrate that we use as an anticoagulant. The ringer lactate isn't permitted as per description from the, what's it called, the firm that's doing the ringer lactate. The information of the ringer lactate states clearly it isn't allowed to use it with the citrate because of side effects. So if there is also any case of a donor adverse event like a hypovolemia event, then we also have a ringer lactate to add afterwards, including medication that that changes the blood pressure by going upwards. So if a donor doesn't have any side effects, then he only requires to get the saline infusion. If there is an adverse event, they get the saline infusion and afterwards the ringer lactate, so they do get stable. We also have a time period after the donation in place, which is in Austria required by law, that they stay up until 30 minutes in the plasma donation center to catch any kind of events that are afterwards presenting to the donors to assure their safety.
I have a question. Dominika Misztela, PPTA staff. Perhaps a question for Michelle Franson and Dr. Darabont. Could you perhaps comment on the value of a donor adverse event recording standard that is specific for plasmapheresis? Because certainly in the SoHO Regulation Proposal, the monitoring of donor adverse events is key, but obviously SoHO substances of human origin differ. So could you perhaps comment on the value of having a standardized system in place, or the audience? Thank you.
Thank you, Dominika. I think the value for having the standardized or the standard is so that every single company or every single donor who donates in an IQPP certified center, the classifications for an adverse event are the same. So if one donor has specific symptoms at one center, it will translate over to another donor having specific symptoms at another standard or another center.
Thank you. My idea to it is very important to speak the same language over all the industry. So we have one language to classify, to grade, to define any kind of adverse events. Also in the EU, up until several years from now, there was no need to report any kind of donor adverse events, only if the quality of the plasma protein was compromised. So the reporting process will start soon. In Austria, the entity who is working with us from the state, the AGES, proposed that we also record donor adverse events, as we already do. This is not a new involvement, but their proposal was to use the donor adverse classification for blood donations. So probably, or at least I suspect, it's only my opinion in this case, they maybe not be aware that there is already a donor adverse reporting in place for the industry. So that might be important to reach out to them and to provide this kind of information. Thank you.
Yeah, I also wanted to follow up. I know there were a number of references to the EDQM references, in particular, and whether or not if any more comment on where they indicated about a risk of cancer as well as bone density, whether or not we feel that those particular papers are valid, or if, just some feedback on that, I'd appreciate.
Yes, as I mentioned, we did not understand the commentaries or the assumptions they made about malignancies and plasmapheresis, and as well as they were mentioned, there might be a harm for the bone in frequent plasmapheresis. To the best of my knowledge, these data were not are not an evidence-based. Are not evidence-based. That's the point. There are some studies focusing on these issues, for instance, for the bone mineralism, but you have to look for the subgroups. For instance, the ages, were there women in the study in the pre or post-menopausal stage, and so on. They're very important information for these issues, and they were not analyzed. And I guess also that the number of donors or was not high enough to prove the impact on it.
Thank you. Dr. Weiss?
Yeah, Rod Weiss from Takeda. I have one single question, maybe to Dr. Darabont and Dr. Mock. As Dr. Darabont, you mentioned the reporting requirements to AGES in Austria. Are you maybe aware of the reporting requirements of donor adverse events in Germany and other countries in Europe?
Okay, that's a very important issue. We are discussing this for years. In Germany, it's not mandatory. We do it on a voluntary basis, and that's really an important point. For some years, there was an hemopheresis registry funded in Germany by the German Society of Transfusion Medicine and Immunohematology, and there was also a very nice paper published in Transfusion Medicine and Chemotherapy about these issues with a lot of blood donation services participating. But now this funding was stopped, and the reporting of and the collection of the data is very difficult, of course, because it's done by the centers, and that's, of course, possible bias. That's right. Austria?
For Austria, of course, we report the donor adverse events, which are severity grade 3, which means major medical intervention as well as hospitalization. That is our requirement nowadays, and also for the whole Europe, up until 2019 and 2020, the reporting of donor adverse events during apheresis donation is only voluntary. The data was not provided only by, I think, 27 countries. The last report I read, that was the SARE paper from European Commission addressing the hemovigilance reports for blood donation, cytopheresis, plasma donation apheresis, and transfusion medicine. So their idea was they acknowledged the need to collect that data for donor adverse events too. So this is a process that will come up in the EU and will be part of our reporting system in the future too.
All right, well, thank you. Before we break for lunch, sponsored by Paysign, there's two quick things. First, I'd like to thank the panelists. Great job with your presentations. Hopefully you all learned a little bit. And so if we could just thank the panelists right now. And then I'd like to invite up Josh Penrod, Senior Vice President and Head of Global Plasma and Technical Advocacy. Got to work on that title, man.
Thank you very much, Roger. This is a previously unscheduled part of the meeting, but didn't know if we could get the picture up of someone that we lost. One of the things that I've learned in my career, and this is my 20th anniversary at PPTA this month, is that after all that time, you get to meet a lot of new people and make a lot of new friends. Unfortunately, too, that also means that sometimes we have to say goodbye. And unfortunately, earlier this year, actually last week, we lost a great icon of the industry, Marilyn Rosa Bray. We had the unfortunate news that she passed after a long illness, and in so doing, we lost a true friend, role model, and leader, and we saluted her here at the conclusion of a panel focusing on donor health, considering that was so critically important to her personally. Now, I've known Marilyn my entire time at PPTA, and she was, of course, a close colleague and a close friend. So I think that probably everybody could recognize then that what I would say would be highly biased, because she helped us so many times, so often, with so many difficult and critical issues over the years, where she contributed in just an enormous and meaningful fashion. But what I can say instead, rather than just my own words, I can take some words that were listed for her from the LinkedIn post which announced her passing. Some of these, I think almost all of you have met Marilyn at some point or another if you've spent time in the industry. You saw her name flash up a couple times during the panel as a co-author or head author of several of the donor health studies that the industry has conducted. But some of the words from friends and colleagues on LinkedIn: iconic, strength, courage, authenticity, compassionate, a woman of character and great worth, kindness, wonderful human being, and one of a kind. And I think that's a legacy that we can all appreciate, and a legacy that we can all strive for in our own lives and our own careers. So, in salute and farewell to Marilyn, I'd ask before we break for lunch to have a brief moment of silence. Great. Thank you, everybody. Have a good lunch, and we'll see you this afternoon. And thank you, Marilyn.